Hello.
Well,
Hello.
Ravi.
Yes. Can you hear me?
Yeah, I can hear you.
Okay. Yeah, it was supposed to be biology today, right?
Yeah. And I'm sorry for yesterday's class. It was an emergency, otherwise I would have attended.
Yeah, no problem. Like definitely I mean, you have to be there with your father. It's okay. All right. So mm, let's continue. I think I was at plant nutrition. All right. Let me check until what did I? Mm, that was it. Was this the last page?
Yeah, this was the last page.
Okay. Just a moment. Yeah. Okay. I don't think I started limiting factors, right?
No, you didn't.
Okay. Today, let's start limiting factors. Okay, so what is a limiting factor? So this is the factor that is in the shortest of life, okay? What are the factors of photosynthesis? So if I want to list that, the factors for photosynthesis are light intensity, because obviously we need light for photosynthesis. Another is carbon dioxide concentration, because we also need carbon dioxide for photosynthesis. Another limiting factor is temperature. Why? Because there are certain enzymes working in photosynthesis and they actually work pretty well at a certain temperature. Okay, so these are the factors for photosynthesis. Even pH can be a limiting factor, and as you go to A levels, you would study that the number of photosynthetic pigments is also a limiting factor, can be a factor. So there are a lot more, but I think in your syllabus right now, we have to deal with these three, if I'm correct. Can you check?
Yeah, it was just these three.
Okay. All right, then.
There's another investigate. I think it's different, right? Necessity for chlorophyll. It's not the same thing.
No, by the way, the investigative studies, so the necessity of chlorophyll, carbon dioxide, and everything, those investigations, you actually need to study those as well, okay? Because I don't have your
Yeah.
book, I cannot really see exactly what they have have in there. So I think you should go through them by yourself at least, okay?
But on my book, I don't think all of the experiments are here.
Sorry?
I think they have the book I have. It doesn't have all of the experiments. Like I don't think they have the necessity for chlorophyll. They have carbon dioxide. I have that experiment, and I have the temperature and light intensity too. But the chlorophyll experiment isn't there on my book.
Okay. I'll try to search something online and yeah, we can discuss it afterwards. Right now, I think let's deal with limiting factors first. Okay. So when we are investigating about, let's say, light intensity, okay, the first one, then we actually have to keep the other factors constant, okay? So every time we change a factor and we want to investigate the effect of one factor, we will keep the rest constant. So if you want to investigate the effect of temperature increase on the rate of photosynthesis, then you definitely have to keep the rest. So these all constant, but this should be changing, okay? So that's how we study the effect of a factor. All right, for example, I can say that, you know, at a constant temperature and carbon dioxide concentration, the rate of photosynthesis increases with increasing light intensity. So like I said, we have to keep two of them constant. And then if we keep temperature and carbon dioxide concentration constant, and then if we increase the light intensity, the rate of photosynthesis will increase as well, okay? But until it reaches a plateau, so it will actually become constant eventually. Why? I'll explain that in a minute, that at that point, either carbon dioxide concentration or temperature or both are the limiting factors, okay? So you'll understand that in just a moment. So when the plateau is reached, light is no longer the limiting factor. Because, you know, it is supplied enough. So right now, the concentration of carbon dioxide or even temperature becomes the limiting factor. Now, when, yeah, at that point, if you increase the concentration of carbon dioxide, only then the rate of photosynthesis will increase again. And then again, there will be a plateau, and then you have to increase increase the temperature. So every time a different thing is a limiting factor, okay?
Both carbon dioxide and temperature can become the limiting factor together, right? At the same time.
Yeah, so I'm going to explain how. So for example, this is, let's say this is the light intensity. Okay. Now, let's say here we have, I don't know, 0.04% carbon dioxide concentration. And let's say we have 25 degrees Celsius temperature. Okay. So you see the rate is increasing up to this point, and then it actually became like a constant. Okay. It reached the plateau. It means from this point, light intensity is not a limiting factor anymore, because it has reached its point, like it's enough for photosynthesis. Right now, either carbon dioxide or temperature is the limiting factor. Okay. So if you, let's say you want to change the carbon dioxide concentration to 0.4%, and the temperature is the same, then you'll see that the curve goes like that. Right here, carbon dioxide is no longer a limiting factor. Why? Because it's not affecting the rate of photosynthesis anymore. It reached a plateau. Right now, definitely temperature is the limiting factor. So let's say we want to see the effect of a higher temperature. So like 0.4% carbon dioxide, but let's say 35 degrees Celsius. Then you'll see that it will increase a bit more. I hope that's clear.
Yeah.
All right. Yeah, that's it. Oh, I don't have enough space. Okay, once you're done, let me know.
I'm done.
Okay, yeah, I'm going to repeat this line again, just to the next page. What do I mean by this line? Is that, let's say we have this graph, rate of photosynthesis by the temperature. Okay. Now, you'll probably see that now you have to remember that in this case, we're varying temperature, so we will keep our light intensity and carbon dioxide concentration constant, okay? So let's say this line is for low light intensity. And this line is for high light intensity. You know, when you keep the light intensity low, but obviously constant, you will see that even if you change, even if you increase the temperature, the rate of photosynthesis is not really enhanced, you know, it doesn't increase that much. Why? Because light intensity is quite low. Light intensity is more, you know, needed for rate of photosynthesis rather than the temperature. So যদি লাইটই কম থাকে টেম্পারেচার ইফেক্টটা কোনো কিছু যায় আসে না বলতে গেলে। But when there is high light intensity, when you increase the temperature, you can see that there's a dramatic increase in the rate of photosynthesis. Okay.
Instead of light, like, is light intensity the most important part for photosynthesis compared to carbon dioxide and temperature?
Yes. Now, one thing to clear out is that don't be fooled. This graph that I showed you, this only shows like half of the picture, okay? This is just showing a small, I don't know, range of temperature. For example, let's say 20 to 40, for example, okay? In fact, if you actually draw a big graph of rate of photosynthesis by temperature, if you see the entire curve, it will look something like this. Why? Because in photosynthesis, there are, you know, a range of different enzymes involved. And at high temperatures, almost all of them would be denatured. Okay. This curve, let's say, is at a 0.03% carbon dioxide and fixed light intensity. So those two are kept constant.
Most enzymes work best at the room temperature, right? Like 37 degrees.
Yeah, that's why you see, you don't see a sharp peak over here. You see like a, I don't know, a blunt peak.
Yeah.
So this means that, there are multiple enzymes having, working their best at these temperatures. Okay.
I'm done.
Okay. Now, if you're confused between this graph and this graph, you could say that this part is what I showed you in this graph. Okay. So, for example, if I increase the light intensity, because it's a fixed light intensity, if I increase it, maybe the curve would reach something like that. Okay. So a higher rate of photosynthesis. I hope that's clear. You don't have to note down, just try to understand everything.
I'm done.
Okay, great. What else? What else? What else? Yeah. So the effect of nitrate and magnesium ions, I think. So there are certain minerals that plants really need for their growth. One is the magnesium ion. Another one is the nitrate ion. Okay. So what do they need magnesium for? making chlorophyll. So if they have a lack of magnesium, you'll be able to see that the leaf will turn yellow. And nitrate ion is needed to make amino acids for protein. And that's why if they have a lack of nitrate ions, they will have no growth. Now, how do you provide them, these minerals? Usually the root takes in these minerals from the soil using active transport. Now, if the soil, has a lack of these minerals, obviously you have to use fertilizers. I think, I'm not sure, but yeah, I think there are certain fertilizers which also add magnesium ions to the soil. But yeah, that's all you have to know, is fertilizers. What type of fertilizers? Such as ammonium nitrate or ammonium phosphate. Okay. This will increase the crop yield. Crop yield means yeah, more crops or more plants which has which have better growth and better health. That's it.
I'm done copying.
Okay. Now about the investigations. Let me see. Mm. Yeah, to actually test for chlorophyll, you need to test for starch. Actually got a page which shows, all the investigation. I'm going to open it. What is it again? Investigation. Save my exams, this one. Yeah, I think they have a list of all the experiments that you need to know. So you can go through them by yourself, I guess. Yeah, so you can test for chlorophyll, light, and carbon dioxide. So starch production and need for chlorophyll. Although plants make glucose in photosynthesis, leaves cannot be tested for its presence as the glucose is quickly used, converted into other substances, and transported or stored as starch. Starch is stored in chloroplasts, where photosynthesis occurs. So testing a leaf for starch is a reliable indicator of which parts of the leaf are photosynthesizing. Leaves can be tested for starch using the following procedure. A leaf is dropped in boiling water to kill the cells and break down the cell membranes. The leaf is left for 5 to 10 minutes in hot ethanol in a boiling tube. This removes the chlorophyll, so color changes from iodine can be seen more clearly. The leaf is dipped in boiling water to soften it. The leaf is spread out in a white tile covered with iodine solution. Okay. In a green leaf, the entire leaf will turn blue-black as photosynthesis is occurring in all areas of the leaf. This method can be used to test whether chlorophyll is needed for photosynthesis by using a variegated leaf, one that is partially green and partially white. So yeah, that's how you can check for chlorophyll. The white areas of the leaf contain no chlorophyll, and when the leaf is tested, only the areas that contain chlorophyll will stain blue or black. The areas that had no chlorophyll will remain orange or brown, as no photosynthesis is occurring here, and so no starch is stored. So these are the steps. Um,
Apu, this
yeah,
starch test, it's only for plants, right? There's a different starch test for animals.
no, I think that's only for plants.
Yeah.
Okay, maybe you can take a picture of these steps, or yeah, you can note it down if you want, but yeah, you can always visit this website.
Mhm.
So there's also the experiment details for need for light to prove that light is needed in photosynthesis, and then also for carbon dioxide. Okay. Okay. Yeah. Do you want me to read through them and explain it to
No,
you, or
Apu, I got the website. I'll read it.
okay, great. Oh, I can download PDF. Wait, really? Okay, no, I don't want to be a member. Anyways. Is it already 14 minutes?
In our syllabus, they ask this question, explain why most farms on life are completely dependent on photosynthesis. There, do we just write about how photosynthesis, like plants are, we are dependent on plants because they make us, like they provide us with food. So we're like,
Yeah. So if they ask you that question, then yeah, you have to first of all say that they provide oxygen through photosynthesis, and obviously we need that for respiration. Like every animal needs that. Yeah, that is the first one, and also secondly, because of starch and because of food. Yeah. So the obviously herbivorous animals, they completely depend on food for plants. They eat plants directly, but also the animals which are omnivores, sorry, which are carnivores, so which actually eat animals, they're also indirectly dependent on the plants, right? Because the herbivores that they eat, they also survive because they eat plants. So I think you have to answer something like this, but I'm not sure about, I mean, how does the marking scheme actually divide the points in this question? Maybe you will get a better picture if you look at the marking scheme for that question. Okay. Yeah. Yeah, I think we will encounter these questions when we do question papers. All right.
Oh, I had another question with point number G. I understand that chlorophyll traps light energy and converts it into chemical energy for the formation of carbohydrates. Would you explain that point?
Which point?
It's in our syllabus number G.
Oh, in your syllabus. Wait,
Yeah.
just a moment. Number G, understand that chlorophyll traps light energy and converts it into chemical energy for the formation of carbohydrates and their subsequent storage. Yeah, so I mean, in light dependent stage, I think I mentioned that, obviously light is needed to split water into hydrogen and oxygen. So yeah, these light energy, this is definitely trapped by chlorophyll. If chlorophyll is not there, this light energy cannot be used. So yeah, so chlorophyll traps the light energy, and eventually you saw that this energy, so this energy is used to split the water, and also later on in light independent stage, I think we used that hydrogen and something else to convert carbon dioxide into carbohydrate. Yeah. So they just want you to explain that and understand this. So yeah.
All the experiments are in that Save My Exam website, right?
Yeah,
I
yeah.
could just
I
read
think
that.
yeah.
Alright.
Yeah, so in point number B, I think they mentioned investigate the necessity for chlorophyll, light, and carbon dioxide for photosynthesis. Yeah, those experiments are in Save My Exams. Yeah, using appropriate control. So go through them. I think that will be enough. And number D, I already taught you the effect of varying light intensity and carbon dioxide and temperature. So these are all about the limiting factors.
Mhm.
Okay, then we can start transport in flowering plants.
But are we going to do animal nutrition later?
Yeah, I think it's good to finish these two together.
That would be helpful for me too.
Yeah, okay. Water and iron uptake, transpiration and translocation. Okay, I think let's first talk about the transport vessels.